Okay. So today we discuss the. Is it clear?
Yes, sir.
Okay. Thank you. So today we discuss the bacteria. So what is the bacteria here? Means the bacteria is very the basic physical unit of the all living organism called the smallest unit capable of life. Its material composition the same in all organisms. So bacteria, which one is belongs to the prokaryote group. Prokaryote means all living organisms divided into prokaryotes and eukaryotes. So what is the prokaryote? Means the the nucleus, which is covered by the nuclear membrane. That one is the eukaryote. In the absence of the nuclear membrane, that one we call as a prokaryote. Here in bacteria, there is no nuclear membrane, means nuclear envelope is absent. So the genetic material only in the form of a thread or a filament-like structures are present. That one we call as the bacterial genophore. Okay, they are reproduced by the asexual sexual methods are there, but the sex organs are absent. Okay, in the bacteria structure, the cell structure of the bacteria, we can see that one. The bacteria, which one is the four, well, five structures are there. Five structures are there. That one is the nucleoid, means the DNA, ribosome, cell membrane, cell wall, and surface layer or slime layer. That one we call as the surface layer or slime layer. Okay. So, hello, is clear? Clear my voice? Kalyani. Hello.
Yes, sir. Clear.
Yes,
Okay.
sir. Yes, sir.
Thank you. So all of you BZC only.
Yes, sir.
Yes,
Thank you. Botany class.
Yes, sir.
Here? Right.
yes, sir.
Right, right. So, so this one is the is five compounds are there. Components are there. That is the nuclear, ribosomes, cell membrane, cell wall, and surface layer. Nothing but we call as a slime layer. So bacteria, which is displays a widely diversity of the shape and sizes are there. Okay. So most bacteria species are either spherical, that one we call as a coccoid, or rod shaped, that one we call as a bacilli. So the spherical structure is there, that one we call as a coccoid. If rod shaped bacteria, that one we call as a bacilli. So elongation is associated with the swimming. Some bacteria are called as a vibrio. That one is the slightly curved rod bacteria are there. So some spiral shaped call are there. That one is the spirilla, or tightly coiled. That some bacteria are there. That one we call as a spirochete. So based on their structure here, the different shapes are there. A small number of these species, even they are tetrahedral, uh, coccoidal shapes are there. Okay. So this one is the bacterial structure. The cell shape. The cell shape is generally characteristic of the given bacterial species, but can vary depending on the growth conditions. The bacteria generally forms a distinctive cell morphologist when they examined by a light microscope. The above structural characters of the bacteria in their small size. The small size is extremely important because it is allow for the large surface area to volume ratio, which allows for the rapid uptake and intracellular distribution of the nutrition and the excretion of the cell wastage. Okay, so what are there here? Some structures are okay. The another one is the bacterial cell wall. So bacterial cell wall, the cell envelope is composed of the plasma membrane and cell wall. The bacterial cell wall provides the structural integrity to the cell. Bacterial cell wall they give the specific shape of the bacteria. Okay, so the bacterial cell wall is differs from that all the organisms by the presence of the peptidoglycan, which is located immediately outside of the cytoplasmic membrane. So peptidoglycan is the specific composition you can identify it in the bacterial cell wall only. Okay, so some organisms are there, special organisms are occurred, their functions and chemical compositions. We can see here the flagella. The flagella, which one is a thread-like structures are there, mainly involved in the swimming movement and which is contain the protein compound. And the pili, now that pilis are the here two types are there, sex pilis and common pilis are there, or fimbria that one. The sex pili mainly involved in the DNA transformation during the conjugation time. Okay, so each one also protein made up of compound is there, and common pili, which one is attached to the surface or the host. This is also the made up of the protein and the capsules are slime layer. Capsules which include the slime layer, slime layer we call them. So which is attached to the surface to protect against the pathogenic um, phagocytic engulfment and occasionally killed. Okay? So which is the polypeptide or polysaccharide compound. So here cell wall. So here based on the cell wall structure, cell wall composition, the bacteria they are divided into gram positive and gram negative bacteria are there. Okay, gram positive. Okay, gram positive and gram negative bacteria. So gram positive bacteria the prevent the osmotic lysis of the cell protoplast and confer their rigid and when they shape on cells. They give the rigid characters are there, which is the made up of the peptidoglycan and murines also present. In gram negative bacteria, peptidoglycan prevent the osmotic lysis and confirm the rigidity and shape. So outer membrane is permeability barriers are there. Okay. So it can also the phospholipids are present, peptidoglycan and phospholipids are there. Protein liposaccharides also present. Okay. So some other organisms are there, plasma membrane, ribosomes, some inclusions also present. Plasmids. This is the plasmids is a special characteristic observation in the bacteria. This is the so this one is the external chromosome genetic material, additional material. This one here we can see the additional material. Okay. So mainly involved these plasmids are mainly involved in the genetic transformations additional material, mainly involved in the biotechnological or plant breeding technique. Okay, so are you clear?
Tell me once again, sir.
Okay, okay. So plasmids are the extra
Not audible,
chromosome,
not
external
audible.
material is there, extra chromosome material. Okay, So this mainly involved in the biotechnological some concepts, means genetic material, plant breeding techniques. So this is the additional material. Okay. So another one is the bacterial cell wall, and this composes the plasma membrane and cell wall. The bacterial cell wall is provide the structural integrity to the cell, bacterial cell wall. Okay. Then based on the gram positive and gram negative bacteria, cell composition is different. The gram positive cell wall or the thick and peptidoglycan layer constitute almost 95% of the cell wall. The gram positive bacteria take up the crystal violet dyes and stain in purple color. Okay, so which is made up of the peptidoglycan, and when we add the crystal violet dyes, so the bacteria has to observe this stain to looks a purple color. If the gram negative bacteria, they are the contain a thin peptidoglycan and which is the pink color. They are, if we are stained, we give this, they are observed and stained as a pink color. So based on this one, here gram positive and gram negative we are clearly uh, diversified. So next one, the nutrition and growth of the bacteria. So we know that one. For the growth and nutrition of the bacteria, the minimum nutritional requirement of water as a source of carbon, source of nitrogen, some inorganic salts. We know that one. So here, some bacteria are the bacteria can be classified based on the nutritional basis, their energy requirement and the ability of the synthesis essential metabolites. Based on the bacteria is divides, sunlight is called the phototrophs. So the energy is obtained from the sunlight, that bacteria is called as a phototroph. Those that earn energy from the chemical reactions, if they are obtained from the chemical reactions, that one we call as a chemotroph, phototrophs and
Sir,
chemotroph.
will you please repeat energy obtained from sunlight?
So that
Please repeat,
bacteria,
sir.
okay, the bacteria which are the energy obtained from the sunlight, that bacteria we call as a phototroph bacteria. Okay, those bacteria are obtained from their energy from the chemical reactions. That bacteria are called the chemotroph. Okay, phototropic bacteria and chemotrophic bacteria are.
Chemo. Okay, sir.
Okay, so those are the synthesis their material, organic material. That bacteria are called autotroph. Okay.
Okay, sir.
Now, so some bacteria are unable to synthesis their metabolites, means food product. So they are depend on other. So that one we call as heterotrophs. Okay. So bacteria are require a supply of inorganic salts also, some phosphate and amino acids, sodium, potassium, magnesium is also they are one. Okay. So growth and multiplication. So bacteria is divided by the binary fission only. Bacteria are reproduced mainly in the binary fission. When the bacterial cell reaches a certain size, it is divided to form two daughter cells. So maturity stage, the daughter cells they can divide and to produce two daughter cells. The nuclear division is followed by the cell division. The interval of the time between the two daughter divisions, mm, the time is required for the bacterium to give rise to the two daughter cells under the optimum conditions is called the generation time. Okay, so based on the optimum conditions only, means the temperature and the requirement of the nutrition. So based on that only, the bacteria can be divided to produce the two daughter cells. Okay, so the bacterial growth curve you can see in the suitable liquid media. It incubated for the growth. Different sources are there. The following the here 4 phases are there. In the bacterial growth curve, here 4 phases are there. That is the log phase, and the stationary phase. So
Phase of
finally,
decline.
phase of the decline means stopping stage. Okay. So first one is the log phase. This initial period is the first period. This is initial period. Time is required for the adaptation of the new environment. The necessary some enzymes, metabolites, intermediate are built in the adequate quantities for the multiplication to the process. Okay. So next one is the log phase. This is the exponential phase also. We call that one. The cells are start to divide in their number and to increase the exponentially. Okay. So another one is the stationary phase. After a varying the period of the potential exponential growth, the cell divide stop due to the depletion of the nutrition and accumulation of the toxic products also. So if the logarithm of the variable count is the plotted against time, a straight line will be obtained. Okay. So another one is the stationary phase. After a very good period of the exponential growth, the cell divide stop due to the depending on the nutrition and accumulation of the toxic products. The number of new cells formed in the just enough to replace the number of cells which are died. Another one is the decline phase. So in this decline phase, the population is decreased due to the cell death. So besides the nutrition, exhaustion, and toxic accumulation, the cell death may be also caused by the autolytic some enzymes also there. Okay. So this is the growth rate of the growth or curve rate of the bacteria. So another one is the bacterial movement. So bacterial movement, we know that one. The bacteria can move using the very mechanism, which are the flagella used for the swimming through the fluids. These bacterial gliding or swimming mortality.
Not clear.
Okay. So bacterial across the surface and changes
It is
the
not clear. It is not clear, sir.
not clear. Okay, okay.
No.
Some disturbance are there. Don't worry about. Again, I repeat. The swimming bacteria is frequently move near 10 body length for the second. Some are the fastest are there. Okay. In the bacterial gliding or switching modality, the bacteria use their pili as the grappling hook repeatedly extended. It is encouraging into the retracting with the remarkable force. Okay. Okay. So the bacterial species is a different number in attachment of the flagella on their surface. There are some flagellas. Some bacteria are having the single flagella. That one we call as a monotrichous. Okay. If the bacteria is having the one flagella, that one we call as a monotrichous. The flagella at each end, that is the amphitrichous. The bacteria we call as the amphitrichous bacteria. If the cluster of the flagella are attached at the a single fold, that one is the low-trichous. Okay.
Okay, sir.
Only the
Low
a group of the flagella are attached at any side of the bacteria. That one we call as a low-trichous. Either the flagella is distributed over the surface of the bacteria, that is the peritrichous. Okay, total entire the bacteria, the flagella is occurred. That one we call as the peritrichous bacteria. Okay. The next one is the reproduction process. In the reproduction process, the bacteria is produced by the mainly here the binary fission and endospores, asexual reproduction and sexual reproductions are there. Okay. So in binary fission, most bacteria rely on the binary fission for the propagation only. Before the binary fission, the cell must copy its genetic material. Means DNA is copied and segregated those copies to opposite end of the cells. Then many types of the proteins and comprise of the cells. The division machinery assemblage at the future division site. Okay. So after that one, the cells will divide and the genetic material also divide. Okay.
Sir, bacteria doesn't have RNA.
So some bacteria, depending on the RNA is also present, some are the DNA is also present. Okay, depending on the species. Depending on
Okay,
the
sir.
species,
Okay.
here some are the DNA, some are the RNA is there. Okay, next one is the endospores. Certain genes, some genera, gram-positive bacteria, Bacillus and Clostridium, those are the highly resistant and dormant structure called endospores are there. There are the resistant from the dot areas and alkalines and some desert conditions, unfavorable conditions, mainly unfavorable conditions, the endospores are formed. So the endospore shows no datable metabolism and can survive the extremely physical and chemical stresses or UV sunlight, gamma radiations, heat. So they are the protect from these conditions, unfavorable environmental conditions. Okay. So another one is the sexual reproduction. In the sexual reproduction involve the fusion of two different cells or transfer of the hereditary factors occur in the bacteria in three different types are there. Okay. So that is the bacterial transformation. So bacterial transformation. Okay. So here the bacterial transformation means the genetic material is transformed. So all of you mute. So bacterial transformation. The bacterial transformation means the genetic material. The bacteria also occur by the transformation, in which the DNA is molecule of the donor cell when they liberated by its designation to take up the another recipient cell, and it's offering inherited some characteristics of the donor cells. When the different strains of the bacteria are found in the mixed state, either in the culture or in nature. So this phenomenon is known as the recombination. So this phenomenon of the transformation was first observed in the grift. Okay, in grift. So in grift, in Diplococcus pneumonia, they are study on the Diplococcus pneumonia. So what happened here? This bacterium causes the pneumonia in males. The grift is found there. Two types of the strains are there. Okay, the pneumonia. So pneumonia, one form the smooth colonies is protected by the capsule, and other one the form the rigged. irregular or rough colonies
Characters carry
without
from
the capsule.
stage to another stage. Means
Okay. So here,
is it right, sir?
what? Hello? Hello? Oh, okay. So when they injected into the moist H chain, only the capsule smooth wall to produce the disease, but the not non-virulent rough cell, that one B. And other hand, when the heat killed capsule virulent, the smooth cell was mixed with the non-virulent rough cell. That one is considered as a D. A, B, and D means A virulent means the capsulated smooth wall cell, which is to produce the disease. B virulent, this is one is the non-virulent and rough. Okay, without capsule wall is there, and non-virulent means they cannot produce the disease. And D virulent means both mixed means capsulated diseased capsulated virus bacteria and smooth cell both are the mixed virulent, which is the non-virulent, that one we consider as a D. So here we are the drift is experimented, that one and were injected in the mice, the disease was produced. When shows some factors of the dead capsule smooth wall and convert into living non-virulent rough cells into the living smooth capsulated cells. Okay, means the genetic material is transformed from the heat smooth to heat converted into non-virulent rough cells to smooth capsulated bacterial cells. So that is the genetic material is transformed. By this experiment, it was shown that the DNA was genetic material is responsible for including the smooth characters of the cell wall, their property of the virulent in moist. This one is the genetic uh, transformation. So another method is the genetic transduction. Transduction. So the genetic transfer in the bacteria is attached to in the process of transduction. Okay. So in Ginder, Ledenberg and Ginder is explained in Salmonella typhimurium. Okay, Salmonella typhimurium, which indicate the bacterial virus or phages is responsible for the transfer of the genetic material from one to other lysogenic or lytic phases are there. Okay, so in this process, the DNA molecule that carries the hereditary characters of the donor bacterium is being transferred to the recipient cells through the energy agency of the phase particles. So in this process, very few closely linked characters can be transferred by each particle. Okay. So another one is the bacterial conjugation. Another method is the bacterial conjugation. So in this conjugation, what happened here? The Jacob was described the conjugation in which two bacteria live side by side for the as much as half an hour. During this period, the portion of the genetic material is slowly passed from one bacterium, which is designated as the male bacteria, to the recipient bacteria, which is designated as the female bacteria. This was established the male bacteria enter into the female one seriously. So here, the bacterial conjugation, the transfer of the genetic material means DNA takes place by the cell to cell contact with the donor and recipient cell. Okay. During this process, the conjugation large portion of the genome is transferred. Okay, this one is observed in the Escherichia coli. Okay, so next one is the economic importance of the bacteria. So in economic importance of bacteria, the bacteria are the unicellular or ultra microscopic organism. We know that one, which is used in the different aspects are there. The bacteria, which are the beneficial aspect, harmful aspect, another one mainly role in the different areas are there. So first of all, we discuss the beneficiary aspects are there. Okay. So what are the beneficiary aspects here? The role in the agriculture. The bacteria mainly very important in the agriculture aspect, the decaying of organic substances. So means most of the bacteria is very useful in the bring out the decomposition of the dead organic matter, means plants and animals. You know that one. The bacteria are saprophytic, that one. They are the decompose the organic material. And to increase the soil fertility, some bacteria maintain those increase the soil fertility. They bring out the physical and chemical changes in their soil, conversion the insoluble material into soluble material. So ammonifying bacteria means those are the bacillus. Bacillus bacteria decaying the dead organic decomposition, the proteinaceous compounds into amino acids. Okay. So here, amino acids are there, so which are reduced to the ammonia by the ammonifying bacteria. So another bacteria are the nitrifying bacteria. So those bacteria which are convert to ammonia salts into nitrates and nitrites, which are absorb the plants. Okay. The nitrifying bacteria are the nitrobacter and nitrosomonas. These nitrosomonas ammonia salts into nitrous acids, which form the nitrates in the salt. Another one is the nitrogen fixing bacteria, for the example, Rhizobium bacteria. So these bacteria take up to the nitrogen from the atmosphere and convert into the organic nitrogen compound. Okay, it is known as the nitrogen fixing. We know that one. So some industrial uses also means preparation of the alcohol and vinegar, butter, geese, coffee, tobacco, and leather industry. Antibiotics also. Antibiotics also they are produced. Okay. So what are there? Antibiotics means the some Alexander Fleming is the Staphylococcus, there is Penicillium.
Sir, I'm looking, sir.
No?
Sir, you are not looking. I mean, class is not coming. Only voice is coming.
Okay, I show some photographs. Wait. Okay.
No, sir, it is not coming.
So here, so harmful, some harmful aspects means um, some harmful aspects also there. That is the which is harmful to direct animals and human beings also, which are the directly or indirectly. There are the cases various diseases in plants and human beings, some domestic animals also. So what are there here? Some animals pathogenic bacteria are there. That is uh, the the tuberculosis. Okay, tuberculosis caused and the mycobacterium tuberculosis caused the tuberculosis disease, and Escherichia typhosa caused the typhoid, and Clostridium tetani caused the tetanus disease in Vibrio, which is caused the cholera disease. like the different bacteria to cause the different diseases are there. And another one is the food destroying bacteria is also there. Clostridium botulism. So Clostridium botulism bacteria and Staphylococcus bacteria to cause the food poisoning. Okay, and soil fertility destroying bacteria is also there, which is very harmful bacteria. These denitrifying bacteria
Sir,
in the
your voice is
soil.
not coming, sir.
Voice not clear?
Voice and your photo is not coming. Textbook photo.
Wait. I will send another pic. Okay.
Okay, sir.
Okay. That is the importance of the bacteria. So
You are not showing anything, sir.
we send that one. Okay. Mm.
Okay.
So here. So soil destroying bacteria, we call as a denitrifying bacteria also. Means these denitrifying bacteria, they are reduced the nitrates and ammonium, so which are escaped into the atmosphere. This process known as the denitrification or denitrifying, which decrease the fertility of the soil. Okay, this one is the some important aspect as well as the harmful aspects of the bacteria. There are caused some common diseases also there. What are the diseases? Black rot disease. So the bacteria to cause some common diseases are there in the different plants or vegetables also. Black rot disease, which is caused the Xanthomonas camprensis. So in the Brassicaceae family members, and bacterial canker means bacterial canker is also Flavibacteria or Michingensis, which is caused in the tomato, capsicum, and chilies plant. Another one is the bacterial soft rot. Bacterial soft rot disease, which is the Pseudomonas species in the all vegetables. Means wide range of vegetables. It include the lettuce, brassicas, and cucurbitous, tomato, capsicum, potato, and carrot, some herbs also. And another one is the bacterial leaf spot disease, which is the Xanthomonas camprensis,
Stress. Yeah.
temperatris, and the ranges of all vegetables. Okay, some vegetables are to cause in the tomato, lettuce, and cucurbitous like that. And another one is the bacterial wilt disease also. This is the Ralstonia solanacearum, which is the in the potato, tomato, capsicum. They are caused. Another one is the bacterial leaf spot or bacterial blight disease, which is the Pseudomonas syringae.
Sir, what is
Syringae?
eggplant, sir?
Hmm? Hmm?
Eggplant means what, sir?
Eggplant, the common names, some common names are there. Okay, eggplant means they are called as a common names. That is the local names are there. Eggplants like that. You should see the common names. Okay, common different. The local names are different. Another one is the bacterial blight disease. So bacterial blight disease also Pseudomonas syringae in the piece, that is the bacterial speak, and which is the
Sir,
Pseudomonas
where are you, sir?
syringae. This
Tell
one
me the
also
page
called.
number, sir.
This one also caused the tomato.
64.
And the bacterial brown spot disease. This is also caused in the beans plants, and the genus is the Pseudomonas syringae, bacterial genus, which caused into the beans plant. Okay. So how to management of these disease? We can see that one. The eradication of the pathogens, we have to remove that one. When the disease is plant, we have to remove from that field and use the clean and seeds and transplant. And another one is the monitoring the crops regularly or used predictive models are there. Okay. So removes weeds and incorporate crop residues when the host diseases are there. And some sprays also means bactericides or fungal side sprays, chemical sprays we are to use. For example, carbendazim or bobistin powders we are used for the control these bacterial diseases. Okay. So the another topic is the next one is the cyanobacteria. Okay. So what is the cyanobacteria here? See here. This one is the bacteria, the bacterial cell.
Sir, voice is
Cyanobacteria.
not coming.
Okay. Now clear? The cyanobacteria means another topic is the cyanobacteria means blue green algae. Cyanobacteria is nothing but blue green algae. Okay. So the cyanobacteria are the aquatic photosynthetic algae. That one we call as a blue green algae. So blue green algae are cyanobacteria because they are the bacteria. They quite small, usually unicellular. Though they are often grows in colonies like structures are there. They are the intermediate bacteria and photosynthetic plants. So here these cyanobacteria are possess the both characters. Here both characters are there means the bacteria characters means prokaryotic characters are there. Okay, prokaryotic characters are there. So that is the they are to related to the bacteria, not eukaryotic characters. So some chloroplast and are present. So that's why they are to produce. They are exhibits the eukaryotic characters. Okay. So general characters are what are the general characters are there in the bacteria? Okay. The bacteria can be found in almost every terrestrial and aquatic habitat. The cyanobacteria include the unicellular or some colony species are there. They form the water columns. Some cyanobacteria they are forming the some vesicles also like structures are there. So they are produced some toxins in the water blooms. That one we call as cyanotoxins are there. The cyanobacteria cultured in the specific media they are to reproduce. Okay, these cyanobacteria is the microorganisms that structurally resemble in the bacteria structure. So that's why we call as a cyanobacteria. So structure of the cell structure of the bacteria means cyanobacteria. So many cyanobacteria form the motile filament cells called the harmogonia that travel away from the main biomass uh, to form the new colonies. Okay. Okay. So here the cells in the harmogonium are often thinner in the vegetative state and the cells on either end of the motile chain by the taperoid. Okay, in each individual cyanobacterium typically has a thick gelatinous cell wall. They are like the flagella, but harmogonia of some species can move about the gliding method. Many of the multicellular filamentous forms of Acetyltorius are capable from the waving motions. The filament oscillates back to and forth in water columns. Some cyanobacteria float by the forming the gas vesicles. These vesicles are not organelles such as okay. They are not bounded by the lipid membranes, but the protein sheath is there. So here in the photosynthetic process here throughout the cyanobacteria do not have a great diversity in the form. So they are very microscopic and they are rich in the chemical diversity. The cyanobacteria get their name from the bluish pigment, that is the phycocyanin or phycoerythrins are there. They also contain the chlorophyll A and some photosynthetic pigments of the plant uses. In fact, the chlorophyll pigment, the symbiotic in the cyanobacteria, okay, phycoerythrin, they form the in the reddish colors are there. Okay. So that is the and reproduction of the cyanobacteria. What is the reproduce? Okay. So here the cyanobacteria are reproduced by the both vegetative and asexual method. Here totally the sexual method is the absence. Okay. The vegetative, the vegetative reproduction form the fission and fragmentation, harmogonia formation, and harmospores and exospores also there. During the asexual method, here during the asexual reproductions are there. There is the aconite formations, endospores, and exospores also there. Okay, so then here, so aconites means special structures are there. That is the aconites. Aconites means they are near in the cells of the heterosis cell. So aconites are very thick
Sir, your
cell
voice
wall.
is not coming properly.
So here aconites now clear. Okay. So
Yes,
actinides here.
sir.
Okay, okay. So actinides are the very thick cell wall formation where in the intercalary cells and near to the heterosis cells. Okay. They are to resistance to the unfavorable conditions. When the unfavorable conditions means they are heat and saltalin are they are protected from the UV lights and X-rays like that only. Okay, UV radiations they are to protect and to when the favorable condition is came, these actinides are germinated and to produce the new plant body. Okay, so another one is the economic importance are there. What are the economic importance means? This is the
Sir, your voice is
actinide.
not getting properly.
Okay, this is the so this one is the actinides. Okay. So some cyanobacteria are sold as a food in the like that. So cyanobacteria is mainly important in the algal biofertilizers. Okay, algal biofertilizers, despite the association with the some toxic in many membrane of the phylum produced. Okay, so another one is the Nasdaq. So so here the Nasdaq. This is the Nasdaq. So the Nasdaq, the phylum is Cyanobacteria, and the class is the Cyanophyceae. Order is the Nastacales. The family is the Nastacaceae. Genus is the Nasdaq. Okay, the Nasdaq common species of the Cyanobacterium in the family of Nastacaceae, which is commonly named in the star jelly like that, and merge eggs and fructice and fascia, uh, which is commonly called. So distribution of the Nasdaq. So the commonly found in the many countries around the world, it is able to survive in the streams. Okay, some streams are there. Okay, they are found in streams and polar regions are there. Some terrestrial as well as the freshwater species are formed from the loose clumps in the soil. Okay, what is the description here? The nasta common in the colony forms of cyanobacteria. It is initially from the small hollow gelatinous globule which grows became the a leathery or flattened or convoluted, forming a gelatinous mass with other colonies growing nearby. Okay, inside thin sheath are numerous and unbranched hair-like structures are there. That one we call as a trichome. So cells have no nuclear or interval membrane system. And to multiply the form of two cells when they divide in binary fission. Along the trichomes, so, mm, the larger specialist nitrogen fixation cells, they are called as heterocytes. They are occurred. Okay. So Nasdaq filaments are composed of a short, rounded to barrel-shaped cells are there. Sometimes undergoing to the fission. Okay. The densely stained vegetative cells contain peripheral convoluted thylakoid with the adequate phycobilins also there. Some carboxysomes, small osmophilous bodies also present in this nostoc. So, mm, so nostoc compound can fix the nitrogen so from the atmosphere. Therefore, they can live in the locations where no nitrogen compounds are available from the substrate. So Nasta common does not have the chloroplast but contain photosynthetic pigments in the cytoplasm of the cell. It also contains the pigments that absorb the long and medium wavelength of the ultraviolet radiations, which are enabled to survive in the places with the high level of the radiations. Okay, what are the uses are there here? Nasta common in the eaten as salads in Philippines and Indonesian, Japan's and Taiwan countries also. The Nasdaq common varieties and flagelliform in the China to form the part of the food traditionally. Okay, they are contain the vitamin C and food stuff. They are the contain. Okay, so the species of the Nasdaq flagelliform and Nasdaq commune are consumed in China, Japan, and some other countries. So Nasdaq commune is also consumed in the some Indonesian countries also, Central Asia, and in Nasdaq ellipsosphorum, that one we contain in the Indonesian. Okay, another one is the Cytonema. So these Cytonema, so the Cytonema here, they are the classification. Phylum is the Cyanobacteria. The phylum is okay. Mm, the class is phylum is the Cyanobacteria. The class is Cyanofaceae. Order is the Nastakales. Family is the Cytonemataceae family, and the genus is the Cytonema. Okay. So here the genus Cytonema is generally considered to be cosmopolitan species. However, which is the contains numerous species which grows only in the tropical and ecological distinctly delineated the habits such as the lateritic soils, dripping rocks, and reservoirs with the water vegetations are there. The Cytonema is the genus of photosynthetic cyanobacteria that contains around the 100 species are there. It grows in the filament to form the dark mats. Many species are aquatic or either free floating or grown attached to the submerged substrate like that water plants are there, while other species grows on the terrestrial rock, wood, soil or some plants. The cytonium is a nitrogen fixer and can provide the fixed nitrogen to the leaves of the plants on which is grows. Some species of the Cytonium form the symbiotic relationship with fungi to produce as a lichen. Okay. So here the Cytonium is a filament form, dark mat or tough among their algae or submerged vegetation in the lakes or terrestrial stones or wood or soil. The trichomes are cylindrical or isopolar and are the usually colored pale green or some brownish is also there. The apical ends of the filament are not tapered, but end of the cells may slightly rounded or winded. The cells sometimes have constricted cross walls and contain group of the long and cylindrical gas vessels are there. So the heterocysts are located throughout the filament are rectangular or solitary with the darkly pigmented cell walls are present. Okay. So the trichomes are contained within the mucilage sheath. They are commonly tied, wide sometimes layer and may color brown and yellow or orange colors are there. So here the cytonema, the specific characters are there. They are the double false branching method. So here double false branching method is there. The filament is breaks apart at the site of the dead cell. So here both ends of the filament then break through the mucilage sheath. They continue to grow as branches. So that's why we can look to like a false branching methods are
Sir,
there.
what is false branching?
So here false branching means the filament breaks apart at the site of the dead cell. They are in the form of the mucilage sheath. So they are covered in the mucilage sheath only. If the dead cells also occur in that place, they are attached with the mucilage sheath is there. So that's why they are looks like a branching like structure, but not a branching. They are attached in the mucilage sheath only. They are so that's why they are we call as a false branching method. Okay. The occasionally false branching is also formed at the heterocyst where the filament breaks apart the similar manner. The several chemicals can limitation blue green also to form some blooms, algal blooms also, algal blooms also. So false branching, hello.
Sir, చెప్పండి.
So false branching are not understood.
Understood, sir.
Okay.
We
So
can
false
understand.
branch. Okay, thank you. So false branching means when the filament cells, where the filament cells are that is breaks down. Okay, at the site of the dead cells. Okay, the some cells are died. We know that some circumstances are there. So some cells are died, and that time that places that site here. What happened here? They are also to attach to the other filament due to the mucilage sheath. Okay, so that's why here they are to form as a branch like structure, but not a true branch. Okay. Okay.
Okay, sir. Thank you.
So this one is the false branching. So in these blue-green algae to contain some algal blooms, also there. Some chemical eliminations are there. That is the copper sulfate, cupric acids, and other Okay, other chemicals also present. Okay.
Sir, explain about Nasdaq, sir, again.
Sorry. Hello. Some chemicals. Several chemicals are eliminated. These blue-green algae in the form of the blooms, algal blooms. Okay, that are these bacteria, means this one. Okay, these cyanobacteria are the eliminate some chemicals also, calcium hypochlorite and copper sulfate, cupric acid. Some chemicals are eliminated. Okay. Okay. So from this one here, the second unit is over. Okay. Okay, clear?
Okay, sir.
Okay, thank you.
Okay, sir.
Okay. So some network issues are there, some network issues. So here some voice is not clear, I think. Okay, the better to connect the next classes. The next class we study the general characters of the algae. Okay. So thank you everybody. Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Sir, good afternoon, sir. Hello, sir.
Good afternoon.
Sir, practicals from when, sir? When are from the practicals?
So practicals are depending on the university decisions only. Okay.
Not from the 7th to 12th, that is not,
Okay.
sir.
We don't know. We don't know about that one. Just frequently visit the website.
Sir, any important questions, sir, please?
Okay, important questions. Last, I will discuss that one.
Okay, sir. Thank you.
So thank you everybody. Okay, regularly you attend the class. Thank you.
Okay, sir. Okay, sir. Thank you.
Thank you, sir.
అదే ఇప్పుడు బ్యాక్టీరియాలో డెడ్ సెల్స్ ఉంటాయని అది ఒకటి తెలుసు అంతే.
బ్యాక్టీరియాలో డెడ్ సెల్స్ ఉంటాయి.
అదే నాకు అర్థమైంది.
First and second block completed.
एक किलो चॉकलेट